Welcome to Study Time, a televised home learning program produced by Rwanda Education Board.
Hello students, welcome to TV Learning Program. My name is Benon. I will be taking Senior 4 students in biology. Students, this is what we are going to do today. Our lesson is from Unit 4 of Senior 4, which is cell structure and specialization. The lesson is transport across the cell membrane. And objectives of our lesson today is explain different methods of transport across the cell membrane and give examples of materials which go in and out of the cell. Before we start our lesson, do you understand what the term cell means? Excellent. The cell is the basic unit of the body, or the smallest structure of living organisms. Do you remember parts of the cell? Yes, we have cell membrane, which is the outermost part of the animal cell. Or we can say cell wall, the outermost part of the plant cell. We also have other structures like cytoplasm, nucleus, mitochondria, ribosomes, lysosomes, rough and smooth endoplasmic reticulums. Those are some parts of the cell. Our concern today will be about cell membrane. Students, why do you think a cell membrane is important to cells? Yes, it is important because it protects the internal parts of the cell. It is also important because it allows materials to go inside the cell as well as outside the cell. Students, here we have the structure of the cell membrane. This cell membrane is a phospholipid bilayer. When you see this part up and there's another part down, that's what we call it phospholipid because it is made up of phospholipids, and bilayer because it is made up of two layers. There's one layer up and another layer down. This cell membrane has carbohydrates. It has a glycolipid. A glycolipid is a lipid which has a carbohydrate. It has cholesterol. It has integral proteins. It has peripheral proteins. It has channel proteins. It has globular proteins. It also has glycoprotein. A glycoprotein is a protein which is made up of carbohydrates. As you see, this cell membrane is made up of many parts. But it is very important because these parts perform different functions. And as we have seen, one of the functions of cell membrane is allowing materials to go inside the cell and materials to go outside the cell. Students, can you give examples of materials or food substances which are needed by the cell? Very good. Glucose is one of the food nutrients needed by the cell. Another example of the substance needed by the cell is oxygen. Very important. We also have lipids which are needed by the cell and other materials. Students, can you give an example of the waste product or waste substance which is produced by the cell and is not needed? It has to be eliminated or it has to pass through the cell membrane going outside the cell? Good. Carbon dioxide is one of the examples. We can also have lactic acid and urea. Those are some of the materials which can go outside of the cell. My question is, how do materials go inside the cell and outside the cell? Can you give methods or different types of methods how materials go into the cell and out of the cell? Good. Diffusion is one of them, and osmosis. Let us discuss one by one. What do you understand when they talk about diffusion? Very good. Diffusion is the movement of materials from the region of high concentration to the region of low concentration through a semi-permeable membrane. Let us take an example here. We have the diagram. This diagram has the cell membrane. Then you can see here we have water, and here we have molecules which are many. Now, as you see these molecules, they are concentrated in one part, and in the other part, there are no molecules. Here, we say that these molecules are concentrated here. There is high concentration of molecules in one place. When they start moving from where they are highly concentrated to where they are less, that is what we call diffusion. The movement of molecules from the region of high concentration to the region of low concentration. Like you can see here, the concentration is high. Here, the concentration is low, which means there is a difference in concentration gradient. For that case, these molecules will move from where there are many to where there are less. This process is what we call diffusion. And it is very important in cells because molecules or particles move from where there are many to where there are less. Or we can even say they move from where they are produced and go to where they are needed. So that process is called diffusion. And which examples of materials that diffuse from one place to another? Here we can talk about gases like carbon dioxide and oxygen can diffuse from one part of the cell or from one part of the body to another part of the body. Example, oxygen. Remember, our body gets oxygen through breathing. And then when we breathe in, this oxygen has to be transported by circulatory system to different parts of the body so that that oxygen can go into cells by the process of diffusion. This oxygen is very important. Can you explain how it is important to cells? Good. It is needed in the process of respiration. And remember, this process of respiration produces energy which is needed by our body as well as our cells, because cells perform different chemical reactions, what we call metabolism, and in that process, there is need for energy. That energy is produced by respiration, which needs energy. And the type of respiration which uses energy is called aerobic respiration. So which means this oxygen enters into the cell by diffusion. When respiration takes place, carbon dioxide is produced. This carbon dioxide which is produced is not needed by cells. It has to go out. And it gets out of the cell by the process of diffusion. Now, after understanding the process of diffusion, which factors affect the rate of diffusion? Which factors can make diffusion go fast or slow? One of them is surface area. When cells have large surface area, the rate of diffusion is high. That means carbon dioxide and oxygen can go in and out easily. Another point is temperature. Temperature is another factor which affects the rate of diffusion. When temperature is high, particles gain kinetic energy. When they gain kinetic energy, they move very fast. And when they move very fast, that means the rate of diffusion is high. Another factor is concentration gradient. With concentration gradient, here it's a very good example. You can see particles here are highly concentrated in one place and they are less concentrated in another place. This is what we call the difference in concentration gradient. When it is there, that means particles can move very fast from where they are highly concentrated to where they are less concentrated. Another factor which affects the rate of diffusion is the diffusion distance. When the distance is short, particles move very fast. And when the distance is long, particles move slowly. Those are some of the factors which affect the rate of diffusion. Can we repeat them? We have seen surface area. We have seen temperature. We have seen concentration gradient. And we have seen diffusion distance. Those are factors which affect the rate of diffusion. Students, do you know any other method which is important and is how materials move across the cell membrane? Yes, osmosis. What do you understand when you hear the term osmosis? Very good. The movement of water molecules from the region of high concentration to the region of low concentration. This is the diagram which is showing osmosis. These blue small particles are water particles, and the green ones are sugar particles. In between, we have a selectively permeable membrane. Remember, one of the traits of the cell membrane is it is selectively permeable. Here, we have high concentration of water molecules. Here we have less concentration of water molecules and sugar particles. With osmosis, water moves from the region of high concentration to the region of low concentration. Water will move from this region to this one. That process is what we call osmosis. Remember, water makes the biggest percentage of our body, which means it is needed by our body cells. So the movement of water from one part of the body to another is what we call osmosis. Students, apart from diffusion and osmosis, which other method do you think materials can use to move from one part of the cell to another or to cross the cell membrane? Active transport. Do you understand what active transport means? This is the movement of molecules against the concentration gradient using energy from respiration. We can say active transport is the movement of particles or molecules from the region where they are less concentrated to the region where they are highly concentrated. Take an example. Here we have a root hair cell. Do you remember or do you know the function of the root hair cell? Good. To absorb water and mineral salts from the soil. Let us say this is the soil, and then here we have a root hair cell. This cell is important in absorbing water from the soil and minerals from the soil. Now, as you see here, we have red particles. Let us say these are minerals. Here we can give example of magnesium. We can give example of nitrates. And when you see those particles, they are highly concentrated inside the root hair cell. And from the soil, you can see they are scattered and less, which means the movement of these particles from where they are less going to the root hair cell where there are many is what we call active transport. The movement of molecules from the region of low concentration to the region of high concentration. Because particles are moving from where they are less to where there are many, there is need for energy from the process of respiration. So active transport is against the concentration gradient because particles move from where they are less to where there are many. Students, here we have seen examples of ions in the root hair cells. Even the uptake of glucose in the small intestine is done by active transport. Even in the kidneys, glucose moves against the concentration gradient and it passes through what we call carrier proteins. So that means there is need for energy because particles are moving from where there are less to where there are many. Students, what are the types of active transport? We have two main types of active transport. One is what we call endocytosis. Another one is what we call exocytosis. How are they different? Here they say endocytosis can be used to bring large particles such as glucose into the cell, which means with endocytosis, particles go into the cell. While in exocytosis, particles move from the cell and go outside the cell. Can we give examples of particles which go into the cell? And they are large particles. Glucose, that is very good. And even ions, like sodium ions, potassium ions, calcium ions, and chloride ions move into the cell by the process called endocytosis. Students, can we give examples of materials which go outside the cell by the process called exocytosis? Excellent. We can have waste products produced by the cell. We can have proteins produced by the cell. Do you remember how proteins are produced in cells? Yes. Cells have what we call rough endoplasmic reticulum, which has ribosomes attached, and then that is where protein synthesis takes place. That's where proteins are synthesized. Examples of proteins here, we can talk about enzymes, which are biological catalysts that speed up chemical reactions in the body. We can talk about hormones, like insulin and glucagon are produced by cells during the process of protein synthesis. Those ones may need to go outside of the cell, going to the parts of the body where they are supposed to produce or to do their effect. So the process by which those proteins, hormones, and waste products get out of the cell is what we call exocytosis. Exocytosis. Which means endocytosis, materials or food substances go into the cell, while exocytosis, materials, substances, or waste products get out of the cell. Students, here we have the diagram showing endocytosis and exocytosis. Like you can see, this green part is inside the cell because you can see the cytoplasm, and this part here, which is extracellular, this one is outside the cell. And you can see here we have substances or solid particles. Here, they are surrounded by the cell membrane and they go inside the cell. This process is what we call endocytosis. And then here we have cytoplasm, we have cell membrane, and we have said food substances or waste products produced by the cell can get out of the cell. That process is called exocytosis. Like you can see here, we have the vesicle, which is going to get close to the cell membrane, and materials can go outside the cell. We have given examples where we have seen some hormones produced by cells can go out. We have seen some proteins, like enzymes, can go out. Even some waste products produced by cells can go out by the process of exocytosis. Students, endocytosis can be divided into two types. We have what we call phagocytosis. We also have what we call pinocytosis. How are these types of endocytosis different? When we say phagocytosis, that is what we call cell eating. And when we say pinocytosis, that is what we call cell drinking. Which means pinocytosis brings liquids into the cell, while phagocytosis is responsible for transporting large solid particles into the cell. We have seen two types of endocytosis: phagocytosis and pinocytosis. Phagocytosis is about cell eating. Pinocytosis is about cell drinking. Let us take an example of phagocytosis in our body. Students, remember, our body has what we call immune system. Do you know the function of immune system? Protect our body from diseases. That is very good. Which means this system is very important. It has different cells. Here we can talk about white blood cells. And white blood cells are divided into other cells, where we can have neutrophils and monocytes. Here, these cells engulf bacteria and viruses by the process of phagocytosis, which means when they engulf bacteria and viruses, then they can destroy them using enzymes. Another example is some large food particles can be engulfed by the process of phagocytosis. Then other solute molecules which have dissolved in solvent, form a solution, can enter into the cell by what we call pinocytosis. Here we have diagrams describing phagocytosis and pinocytosis. Here you can see we have a very large substance. Here we can even say bacteria, and this bacteria can be engulfed by our body cells like neutrophils by the process of phagocytosis. And then here we have dissolved solutes which enter into our cells by the process of pinocytosis. Students, let us make a quick review of our lesson today. We have seen the structure of the cell membrane. We have seen that the cell membrane has different functions. One of them is allowing materials which are useful to go inside the cell, and also to allow materials to go out of the cell. Some of those materials can be useful, like proteins and hormones. Some can be waste products produced by the cell. We have seen that there are different methods through which materials can go in and out of the cell. We have seen diffusion, we have seen osmosis, we have seen active transport, and active transport, we have seen that there are two types: endocytosis and exocytosis. We have also seen that endocytosis is divided into two other types: phagocytosis, which is called cell eating, and pinocytosis, which is called cell drinking. Before we finish our lesson today, let us have homework. The first question is, define the term diffusion. Explain factors which affect the rate of diffusion. Number 3, what is the difference between diffusion and osmosis? Explain how large molecules cross the cell membrane of the cell. Number 5, differentiate endocytosis and exocytosis. Number 6, how is phagocytosis different from pinocytosis? And the last question, explain the process of phagocytosis in macrophages, neutrophils, monocytes, and Kupffer cells. Kupffer cells are cells we find in liver. Thank you very much for today. Enjoy the rest of the day.